Collaborative Seminar Series. I am Lynne Maquat, and I head the Center for RNA Biology at the University of Rochester. Welcome. We have two speakers from Rochester today, and I am going to let Dragony Fu introduce our first speaker. But before I do that, you will see that there is a link on several of these slides to Discord, and that is what any of you can join at 5 o'clock to speak further to the seminar speakers if you haven't been able to ask any questions that you have of them in the Q&A session after each talk. The talks are generally 20 to 25 minutes. We'll have 5 to 10 minutes of questions and answers, and then there's the time at 5 o'clock to ask more questions and get more answers. All right, so Dragony, you will introduce Jenna Lentini.
All right, great. Hello, everyone. My name is Dragony Fu, and I'm a part of the Center for RNA Biology here at the U of R. And it's my pleasure to introduce Jenna Lentini, who is a graduate student in my laboratory. And Jenna Lentini graduated from SUNY Geneseo, where she subsequently joined my lab. And she has made a number of discoveries on the targets and functions of RNA methyltransferases. And so today she'll be talking about one of them.
Okay, thank you, Dragony. So hi, I'm Jenna. I'm a PhD student in Dragony's lab, and I'm going to be talking to you today about my work, which we actually recently just published a few months into the start of the pandemic. So I guess one good thing came out of 2020. But my talk today will be entitled DALRD3 targets specific arginine-tRNAs to facilitate 3-methylcytosine modification. Okay, so I'm not going to go into too much background as we're an RNA audience. However, I do want to point out that RNAs are the most highly modified RNA species. So you can see that RNA modifications can happen throughout the body of the tRNA that can impact its structure, its stability. So hypomodified tRNAs can be subjected to degradation and can also influence protein recognition. So for example, with the ribosome. Modifications can also directly impact translation, where it can affect aminoacylation of the tRNA, as well as impacting decoding, efficiency, and fidelity. And it's come to light recently that individuals who have mutations in tRNA modifying enzymes, thereby lacking these modifications, oftentimes leads to neurodevelopmental disorders. But the mechanism underlying how loss of these modifications leads to this disorder is not really well understood. But we do know that tRNAs have over 100 different chemical modifications that can occur, many of which can just be a simple methylation, which can occur throughout the tRNA, denoted by these little red dots here. And the modification that we're going to be focusing on today is a cytosine at position 32 that can be modified to this 3-methylcytosine modification, which I'll be referring to as M3C in this talk. And this is a eukaryotic specific modification of tRNA. And although we have known about this modification for decades now, the biological impact of it has not been well established. It's thought that position 32, or it's known that position 32 makes non-canonical base pairs with position 38. So possibly this modification helps to stabilize that anti-codon loop and thus possibly could impact translation, but the direct role is not known. But what we do know is that M3C is found on a range of different tRNA species. So if you look at yeast or other invertebrates, you see this modification occurring on 2 different species, so tRNA serine and threonine. And when you look at individuals or organisms like mammals or vertebrates, you again see these 2 tRNAs, serine and threonine, but there's also an expansion to novel substrates. So you then see tRNA arginine as well as mitochondrial tRNAs having this modification. Additionally, other positions, so the variable loop of tRNA serine, as well as 2 other positions on tRNA methionine and leucine. So to us, it seems like this expansion of novel substrates must indicate that this modification is important, but why it's important, we don't know yet. So to really understand these modifications, it's important to know the enzymes that catalyze these reactions. So this was originally done by two independent groups, so Suzuki Lab as well as Eric Fizicki. And Eric Fizicki actually is another researcher at the University of Rochester. So we're really lucky to have him here and be able to collaborate with him. So they found that it was in yeast, this TRIM140 protein that's responsible for modifying the two different tRNA species. And this protein has an N-terminal actin binding domain, as well as a C-terminal methyltransferase domain, which is necessary and sufficient for modification. And interestingly, so in yeast, we have this one TRIM140 protein that we know modifies both tRNA threonine and serine. But in humans, again, there's this diversification of even the enzyme family. So we have 4 different homologs, metal 2A, 2B, 6, and 8. And we know that metal 2A and 2B modify both tRNA threonine as well as this novel substrate arginine. Metal 6 modifies tRNA serine. And it was found that metal 8 modifies mRNA. However, we also found that metal 8 modifies mitochondrial tRNA as well, which I will not be discussing today, but we have a preprint out on bioRxives if you would like to check that out. But it's not currently really understood how the human enzymes are able to efficiently modify these novel substrates. So today we're going to be looking at metal 2A and 2B and their methylation of arginine. So this brings us to our research goals. So overall, we want to understand how the metal 2 in particular evolved to recognize and modify tRNA arginine. And the larger goal that we're trying to solve is explore the biological impact of M3C. So first, we wanted to see what other proteins are interacting with our metal 2 proteins. So we had tagged metal 2 proteins that we created stably expressing cell lines for, extracted out the RNA, purified our metal 2s, and sent the elutions to mass spectrometry. And we were really intrigued when we looked at and saw both metal 2A and 2B were interacting with this uncharacterized protein called DALR, anticodon binding protein 3. So when we looked this protein up, there was no papers that we could see about this protein. But what we did know is it has the C-terminal DALR domain. And this domain is also found in 2 other proteins. So arginyl-tRNA synthetase, as well as the mitochondrial arginyl-tRNA synthetase, both contain the C-terminal DALR domain. These 2 in particular both have a catalytic domain as well that is not found in D3 that allows them to amino acylate a tRNA with the corresponding arginine amino acid. We also know that D3 is a vertebrate specific protein, whereas the RARs and RARs2 are found in all domains of life. So we next wanted to ask, so we know what this domain does. We know that it allows recognition and binding of certain tRNA anticodon loops, specifically of arginine-tRNA. So can D3 affect metal 2A and 2B's ability to interact with RNA? So to look at that, we did a similar experiment as before, where we took tagged metal 2A and 2B, expressed them in human cells, and purified them either with or without D3. And then we looked at co-purifying RNAs. So here's a total nucleic acid stain, where you can see the first few lanes are the input. So you can see a band denoting 5.8S, 5S of ribosomal RNA, as well as tRNA. And you can see when we purify metal 2A and 2B alone, we do see some co-purification of tRNA. So from here, we can use northern probing to actually look at what tRNA species what is in this co-purification. So we can see that they both purify with arg-CCU as well as UCU. It's a little bit hard to see here. And they also co-purify with tRNA threonine, which makes sense, that's a substrate. However, we didn't see any co-purification for tRNA serine, which does have M3C. And we didn't see any for glutamine, which is not an M3C containing tRNA. But what was really interesting is when we co-expressed D3, we saw a enrichment for both of these arginines compared to a subsequent reduction in threonine. So from here, it seems that metal 2A and 2B can interact with both tRNA threonine and arginine. However, to efficiently interact with arginine, it needs to be in complex with D3. So we next asked, is D3 required for methylating tRNA in vivo? So to do this, we created human knockout cell lines using CRISPR. So this was done in human haploid HAP1 cells. So you can see here through immunoblotting for wild type, you see a band here denoting D3 that is absent in the knockout. And to monitor M3C status, we use a primer extension assay. So this relies on a radio-labeled probe that can hybridize your tRNA of interest, and using reverse transcriptase to extend this product. If there's a bulky modification like M3C, it will block the RT from going through, resulting in a shortened product. However, if it doesn't have the modification here, the RT will read through to either the next modification or to the end of the tRNA. So when we look at just positive controlled 293tRNA, which we know has M3C without RT, so if this dark band right here, this is the radio-labeled probe. So without RT, we don't see any extended products. With RT, we see a robust signal for M3C detection here. And then when we look at our HAP1 wild type cells, we again see a stop indicative of M3C formation. However, when we look at the knockout cell lines, we see a reduction in M3C formation as well as subsequent read-through products. So it seems that the D3 knockout has a reduction in modification for tRNA arginine CCU. We also looked at UCU, which I'm not showing you here, but we see the same effect. When we look at threonine, comparing our positive control as well as our wild type and knockout, we did not observe any difference. So this seems specific for arginine tRNAs. We next wondered if there are particular parts of D3 that confers this M3C formation. So in our knockout lines, we were able to transfect 3 different constructs. So we also had a vector control. We have a full length wild type D3, a D3 that lacks the DALR domain, as well as one that was just the DALR domain that lacks the N-terminal portion. And again, we use primer extension to assay for M3C formation. So here in our wild type with our empty vector, we see a M3C formation right here that is reduced in our knockouts. However, when we re-express full length wild type D3 in our knockouts, we see rescue of modification. However, we didn't detect any significant rescue in both of our truncation products. So it seems that both the N-terminus and the DALR domain are required for M3C formation. In the future, we would like to look more specifically to see if there's any specific residues that are important for M3C formation, but as of right now, we don't know of any particular residues. So what about the tRNA itself? So interestingly, there are 5 different human tRNA isodecoders, but only 2 of them get modified, so CCU and UCU, whereas 2 of the other ones do have a C at position 32, they just don't get modified. So when we looked at the anticodon loop, we noticed that in the 2 that get modified, there is a U at position 36, as well as a modified A at position 37. So we wondered, are these 2 residues important for M3C specificity? So to address this question, we took purified metal 2 and D3 complex, incubated it in a test tube with the SAM methyl donor, as well as in vitro transcribed RNA, and again, use primer extension to assay M3C. So what we saw is just normally on normal CCU in vitro transcribed RNA, the metal 2A D3 complex is able to methylate in vitro. So again, we see a robust signal for M3C formation here. And we know this is happening at position 32 because when we mutate the C to an A, we lose this modification. And then when we look at U36 to a G substitution, we again see a reduction in M3C formation, as well as on position A37 to a G. Here's the quantification of that. So comparing the U36 and A37 to the wild type, there is a significant reduction. So it does seem likely that both of these positions do confer some sort of specificity in terms of all 5 of the isodecoders. We also wonder if CCU and UCU, if there's something just inherently unique about these that maybe M3C gives them a benefit and why they have to have M3C. We don't have an answer to that question right now, but we do have some really intriguing preliminary data. So if we look at our wild type and knockout cells that have been rescued either with a vector or full length D3, harvest the RNA and run it on a non-denaturing polyacrylamide gel. And if we probe for arginine UCU, we observe in the wild type alone and with vector there's two distinct bands. However, when we look at the knockout or knockout with vector, we only see one lower band. And then what was really interesting to us is when we rescue with the full length D3, it returns to these two distinct bands. And we don't see this for CCU. So we're not exactly sure right now what this means. It might indicate that there could be two different folding conformations of arginine UCU, that when you lose the modification, you're kind of restricted to just one conformation. But this is something we are really excited to follow up. We don't know what it means right now, but we're really excited about it. So there seems to be differential folding effects on the two tRNA substrates. So this all points to the bigger question of, is there a biological role for D3 and even larger than that, M3C in general? So our collaborators in Saudi Arabia identified a family. So there is a healthy mother and father who are healthy first cousins, and they had 3 living children, 1 spontaneous miscarriage. So 1 child is healthy and 2 children suffer from epileptic encephalopathy. So this is a severe epileptic disorder that has very early onset within a few months of being born. They also have cognitive and behavioral deficits, as well as psychomotor dysfunction. And unfortunately, one of these individuals does not take to anti-seizure medications, so it is pretty severe. And they found that this is a homozygous recessive mutation in D3, causing a premature stop codon, where the mother and father and the 1 healthy child are all heterozygous, and the 2 individuals are homozygous for this mutation. So this nonsense mutation should lead to a truncated protein that would not have the DALR domain, or potentially the mRNA could be subjected to NMD. So using immunoblot, we look to see for D3 expression, and our collaborators made lymphoblastoid cell lines that we were able to work with. And from these cell lines, we were able to detect that in the 2 ethnically matched wild type controls, there is formation, or we do see expression of D3. However, that expression is lost in the 2 individuals with the mutation. And then again, when we assay for M3C formation, specifically looking at arginine CCU, we see in our 2 wild type controls that there is a stop for M3C. However, our 2 individuals that are affected by epilepsy have a reduction in M3C with, again, a pretty pronounced read-through product. So it seems that they are lacking M3C formation on ARG-CCU. We again looked at UCU and saw the same effect, but we didn't see this for threonine or serine. And here's the quantification for that, comparing the 2 patients with the wild types. So in conclusion, it seems that metal 2A and 2B can interact with both threonine and arginine and methylate it. However, to efficiently methylate arginine CCU and UCU, it requires being in complex with D3. And we know that D3 is involved in neurodevelopment because individuals that have a nonsense mutation have epileptic encephalopathy. So we now know that these individuals lack M3C formation in arginine CCU and UCU. So possibly this is the bridge between having a mutation in D3 and then having neurodevelopment. We can't rule out that D3 doesn't have other functions that we're not currently aware of, but this seems like a likely pathway to follow up on, and we're really excited about it. And I just want to end with one last preliminary data that will be the future studies for our lab. So we actually acquired a D3 knockout mouse that was made through CRISPR. So it has a exonal deletion in exon 2 that again should create a truncated protein. And we verified this through immunoblotting. These mice are viable. They don't have any observable phenotype or morphological differences. And unfortunately, many mouse epilepsy models exhibit no major change except when they're challenged with seizure-inducing conditions. So we have not done that experiment yet, but we are hoping to do that soon. But what we do know is that if we're, again, looking at a primer extension analysis between our wild type and our D3 knockout mice, we do see for arginine CCU that there is a reduction of M3C formation. And this is also the case for UCU, as well as the brain-specific UCU transcript. Now, when we look at just steady state levels of tRNAs, we don't see any changes. However, when we look at the brain-specific arginine UCU transcript, that's when things get a little weird. So here, if we're just looking at a northern blot, so this is probed against the brain-specific ARG-UCU, and using different tissue types, you can see comparing wild type and knockout in the liver, kidney, and heart, you see very low background binding signal. But when we look at the wild type in the brain for the brain-specific ARG-UCU, we see pretty high levels that are greatly reduced in the knockout. And just probing for a loading control, a U6 RNA, there doesn't seem to be any issues with the RNA in our knockout. So there does seem to be, in the D3 knockout, there is a decreased level of the brain-enriched tRNA arginine UCU. So possibly this could be explain how these individuals are suffering from neurodevelopmental disorders. And this is something that we really hope to look into in the future. And we're really excited about starting all this mouse work for the future. So with that, I just want to end with the importance of our study. So here we have elucidated a novel mechanism by which specific tRNAs are recognized by a previously uncharacterized synthetase-like mimic. Vertebrates have evolved to harbor M3C on tRNA arginine, and this correlates with the advent of D3 in the vertebrate-specific lineage. And this is the first time, the first physiological role of M3C tRNA modification in proper neurodevelopment. So with that, I would just like to thank my lab, especially my advisor, Dragony Fu. I'd also like to thank Kasia, Ray, and Lindsay and Kyle, who are currently working on the D3 mouse or have contributed to that. Thank you to all of our collaborators at the University of Rochester, as well as our collaborators in Saudi Arabia. Thank you to the RNA Collaborative Seminar Series for allowing me to speak, and for all of you for listening to this talk. And here's our funding sources.
Thank you, Jenna. That's great. So while Todd Lowe said, wow, nice talk, I have a question for you, Jenna, while we're waiting for other questions. Have you looked during embryogenesis, for example, as to when D3 starts to be expressed in different parts of the brain of the mouse?
Yeah, that is a great question. We have not looked into that. Now that we have these mice, we can start doing those sorts of studies, but we don't know right now. That's a great question.
So I was thinking of the wild type mouse. If you want samples during embryogenesis and post-birth from the cortex, cerebellum, and hippocampus for Western blotting, we can give them to you.
Oh, okay. That would be great. Yeah, hopefully we can talk after this then and get that going. Yeah, that would be great. Thank you.
You're welcome. Okay, are there any other questions? Okay, I have another question. So you said that when you have a loss of the M3C32 modification, you think that you have a change in tRNA folding. But do you know if it's folding that, or if it's another modification that doesn't happen to the tRNA or that does happen to the tRNA? You had a kind of strong stop when you didn't have modification that was higher up, and there was a faint band in the wild type properly modified. So what do you think?
Yeah, so that's definitely on our radar. It could be a possibility. We're hoping to use mass spec to look at a whole range of different tRNAs and see if they're affected in our wild type and knockouts. As of right now, since we were comparing our non-denaturing gels with our denaturing gels, if there was a change in modification, you might still be able to see that change on a denaturing gel, which we don't. But we can't say for certain that that's not happening. And that is something that we would like to look into.
Great. Okay, Rich Mariah, how do we hear your question, Rich? He says he has a question, but I don't see what the question is. Rich, do you want to just speak out your question? Anybody have a question? You can just either just ask your question, unmute and ask it.
We have a couple in Q&A.
Oh, I didn't see those. Okay. What are they, Liz? I'm not seeing them.
So Ambro Van Hoof
Okay,
says, what is different about the brain-specific UCU?
So yeah, that's a great question. So this brain-specific UCU, it is really the only difference. So it still has the same modification. It has M3C. It also has another modification that our lab works on, M22G, but it is specifically enriched in the brain. So there is a brain-specific gene that causes this transcript to be there. And it does have some implications already with neurodevelopment. So Susan Ackerman's paper has talked about this in depth. So it's definitely an intriguing transcript. We don't know too much about what's going on yet with our system, but it's an avenue we are really excited to explore.
okay,
There's another question from Pong. I'll read it off for you.
okay.
Do you expect that there will be ribosome stalling at brain-specific tRNA arginine codons on mRNAs such as AGA that may cause translation deficiency?
Yeah, so again, that's another great question, something that we hope to do in the future. We definitely, there's precedence again with Susan Ackerman's paper that might be occurring. We don't know right now, but ribosome profiling is definitely something we're interested in pursuing.
Okay, here's Rich's question. Are there mitochondrial substrates of D3 in human?
We don't know right now. Great question. Yeah, with the mitochondria, I've only focused on mitochondrial tRNA, threonine and serine, and both of those seem to be modified by metal 8. And we don't see any evidence of metal 8 interacting with D3 at all. Mitochondrial arginine doesn't have M3C, so I wouldn't suspect D3 would be involved there. But if it has other functions, I'm not sure.
Mhm. And then we kind of answered Todd's question. The D loop also has two insertions at 20A and 20B. Could they affect folding?
For the brain specific or just in general? I don't see that question, sorry.
Okay, he doesn't specify. Oh, now there's a
I
lot.
mean, that could definitely contribute to
Oh,
some
he
sort
means
of-
brain specific.
Okay, yeah, so that's definitely something else that we can look into. We don't know right now.
Okay, what type of mass spec would you be using to determine M/Z FT ICR MALDI-TOF? What would you be using?
We have a proteomics core that helps us do that. I don't know the machine they are using off the top of my head, sorry.
It's LCMS.
Yeah.
LCMS,
Okay,
LCMS-MS.
how are we doing on time? We have a few more moments. I am not seeing other questions.
Someone asked, is there a human brain-specific tRNA arginine? Jenna?
So yes, that is there is one in humans as well. Unfortunately, the patient cell lines that we have, they are blood cells, so we're not able to characterize that right now. So we're using the mouse model to explore the brain specific.
Why can't you use the, oh, because of their blood. Okay, fine. Okay, well, thank you. That was excellent.
Thank you.
And remember to go to Discord at 5 o'clock and there may be other questions for you there, Jenna. All right. So we're more or less right on time. And I will introduce our second speaker, who many of you already know, Dave Mathews. He is a professor in the Department of Biochemistry and Biophysics, and also biostatistics and computational biology, and of course, our Center for RNA Biology. He is the Associate Director of our Center for RNA Biology, and Dave is also very active in graduate education in the biophysics, structural, and computational biology domain. Many of you know that Dave's research spans computational biology and bioinformatics, and he's very well known for his work determining RNA structure so that RNA can be utilized not only as a tool, but a target for therapeutics. So with that, Dave, take it away.
Thanks, Lynn. So today I'm going to talk about using conservation to determine RNA secondary structure. And so I'll start by talking about how secondary structure is predicted. I'll then talk about how conservation helps us to model secondary structure. I'll talk about the Turbofold approach that we developed. And I'll talk about making Turbofold fast enough for whole viral genomes and some first predictions we made for SARS-CoV-2. So this slide shows the hierarchy of RNA structure. On the left side is the primary structure of a sequence, which is the sequence of nucleotides. In the center is the secondary structure, which is the set of the canonical base pairs. And on the right is the tertiary structure, or the three-dimensional arrangement of the atoms in space. We know that base pairs tend to be more thermal stable than tertiary contacts. And we know that base pairs tend to form on much faster timescales than tertiary contacts. So for those two reasons, we say RNA structure is hierarchical, and we can predict secondary structure without having to predict 3D structure. So my group is best known for our work in taking sequence and predicting secondary structure. We also work on the problem of starting from a secondary structure, modeling the 3D structure. And we also do a lot of work modeling dynamics of 3D structures and conformational change pathways. We also do some work going backwards along this left arrow. So we work on the problem of designing a sequence that will fold to a specific secondary structure. And we use our ability to predict secondary structures to scan genomes to find novel non-coding RNAs. So in this talk, I'm going to focus on the secondary structure prediction problem. And so the minimum free energy structure is the most favorable. So if we let an RNA reach its equilibrium structure, and there's some good reason to believe that RNAs reach their equilibrium structure in cells, then there'll be an equilibrium between the unpaired state and a given structure I. That's governed by equilibrium constant K, which is the concentration of that structure divided by the concentration of the unpaired state, which by definition is E to the negative change in Gibbs free energy divided by RT, where R is the gas constant and T is the absolute temperature. So what that means is the lower the folding free energy change, the more favor a structure, and lower means more negative. Given a secondary structure, we have a good model for estimating the folding free energy change. So we call it a nearest neighbor model, and it's often called the Turner rules, named after Doug Turner in our chemistry department. And so this is an example calculation for a short RNA stem loop. And you can see that the helices favor folding because they have negative folding free energy changes. And in the nearest neighbor model, we look at stacks of adjacent base pairs, and we can look them up in tables to determine their values. Then in this stem loop, there's a terminal AU base pair, and that costs about half a kcal per mole in stability, because that's relative to what a GC end would have. Then there are two terms that are sequence specific for stacking in the loop. And so the nearest neighbor rules need to account for some of the sequence dependence of loop stability. And then closing loops costs an unfavorable increment of free energy. And we call that initiation. That's largely the entropic cost of going from a random coil to fixing to a specific loop conformation, closing that loop. And then in the nearest neighbor model, we sum these increments to estimate the total folding stability. So in this case, we would estimate minus 1.58 kcals per mole. Okay, so how then do we actually predict the secondary structure? So what might come to mind would be just generating all possible secondary structures and ranking them by stability. But it turns out that's naive. And the reason is that the number of secondary structures increases by about 1.8 to the n, where n is the number of nucleotides. So for a 25-mer oligonucleotide, this would work. There's only about 2 million possible secondary structures. And on a reasonably fast computer, we could do this in 40 minutes. But by the time we get to a tRNA of 100 nucleotides, there's about 3 times 10 to the 25th possible secondary structures. And the time to do this calculation would be about 10 to the 15 years. The universe is only 10 to the 10 years old. Likewise, by the time we get to a ribosomal RNA of 1,000 nucleotides, there's about 10 to the 255 possible secondary structures. There are only 10 to the 80 atoms in the universe. And of course, by the time we get to HIV RNA, these numbers are beyond astronomical. So we have a good solution to this problem, and that is to use what's called a dynamic programming algorithm. And what's really exciting about this is that the lowest free energy structure is guaranteed given the nearest neighbor model parameters that are used. And the way it works is that obviously it doesn't explicitly generate all these structures, but the dynamic programming algorithm can implicitly consider them all using recursion. And what that means is we solve the folding problem for shorter sequences and build up to longer and longer sequences. So we don't make fixed choices. We have to consider all the possible structures, but the recursion lets us do that quickly. And Sean Eddy wrote a nice review in Nature Biotechnology explaining how dynamic programming works. So then in summary, to predict a secondary structure, the input to our software is a sequence that we want to predict the secondary structure of, the set of nearest neighbor parameters that estimate the stability of structures, which are called the Turner rules. These then go into a dynamic programming algorithm that solves the search problem exactly. And then we can output our predictions. So they could be predicted secondary structures, or we can predict the base pairing probabilities for individual base pairs. And so this shows the result of a benchmark I did quite a long time ago, so back in 2004. So I assembled a database of RNA families with a large number of known secondary structures. And for these families, I then predicted the secondary structures and calculated the percent of known base pairs correctly predicted. And on average in this benchmark, we were able to predict 73% of known base pairs. So that's good. So it's like a passing grade, but it's not an A. And so we've been working on improving the methods. And so the different ways our group has worked on this is by improving the nearest neighbor parameters. We work on improving the algorithms. We've done a lot of work using experimental mapping data in the structure predictions. So for example, shape or DMS data, and incorporating that into secondary structure prediction. But we also work on predicting conserved structures using multiple homologous sequences. And I'll talk more about that. So comparative sequence analysis is a manual technique that is very accurate at determining secondary structure. And so in this technique, a conserved structure is determined from multiple homologous sequences. So these are generally sequences from different species that perform the same function and are related in evolution. And what supports the structure are compensating base pair changes. And so this is an alignment of 10 tRNA sequences. So each line is a different tRNA. And then I indicate with underlining in brackets the nucleotides that are base paired. And so you can get a quick glance overview that the base pairs are conserved across the 10 sequences. But I want to focus on the anticodon stem loop of the first two sequences. So the first and second sequence both form a stem loop. So this is the first sequence, this is the structure of the second sequence. What's highlighted here is that across evolution, the sequence changed. So in this case, a UA base pair changed to a GC base pair, and a CG base pair changed to an AU. And so we call these the compensating base pair changes. They indicate that the structure was conserved even though the sequence wasn't, and two changes in sequence needed to occur to conserve the structure. So this makes comparative analysis a chicken or egg problem in the sense that the alignment reveals the conserved pairs, but we have to know the conserved pairs to determine the alignment because the sequence is not conserved. And another aspect that makes this problem complicated is that structures can also evolve. So here I show two different types of tRNAs. So generally we think of tRNA as the cloverleaf structure with 4 branches, but there's a large subset of tRNAs that have 5 branches and can perform the same function. So we need to be mindful of these insertions of structures. So to try to automate comparative sequence analysis, we developed a method which we call Turbofold. This was a close collaboration with Gaurav Sharma, who's in our Department of Electrical and Computer Engineering. And we jointly advised 3 students who worked on this project. So Zhen Tan, who's pictured here in a photo of my group from a few years ago, Yinghan Fu, and Arif Harmansi. And so one of the things I learned from Gaurav, who has a background in digital communication techniques and signal processing, is about turbo decoding. So it turns out when we talk on our cell phone, that message has to be sent by radio wave to a receiving tower. And of course, a cell phone only has a certain amount of energy it can use to transmit. So there'll be some noise that'll be picked up in the signal. And so one way to improve the quality of the signal is to split the encodings or split the energy into multiple encodings of the same voice signal. Then at the receiving end, these are robustly decoded to make a voice signal. And it's provable that it's better to split the energy in the two different encodings than to put all the energy into one encoding. So what Gaurav and I realized is that we have something highly analogous with multiple homologous RNAs. They represent multiple encodings of the same secondary structure. So we developed a method we call Turbofold that takes as its input multiple homologous RNA sequences. The output is then estimated base pair probabilities for each sequence, where the sequences inform each other as to conserve base pairs. And the central insight behind Turbofold is illustrated on this slide. So in the upper left-hand corner, I show estimations of base pair probabilities for a tRNA, RD0500. And so in this plot, I have nucleotide index for RD0500 on both the X and Y axis. So a dot here indicates an estimated base pair, where a black dot, like here, indicates a base pair predicted with almost 100% pairing probability. And the lighter the gray, the lower the estimated pairing probability. So there's some uncertainty in the predicted secondary structure in this region of the sequence. We can then take this tRNA RD0500 and we can align it with a hidden Markov model to get the probability of nucleotide alignments to another tRNA, in this case, RD0260. So in the upper right-hand corner, I've plotted the alignment of nucleotides. So the y-axis is nucleotide index from RD0500. The x-axis is nucleotide index for RD0260. So a dot here is an alignment between nucleotides. Again, where black indicates essentially 100% alignment probability, and gray indicates some uncertainty. So in this alignment of the two tRNAs, there's some uncertainty at the 5' end. So with this information in hand, we can then take our estimated pair probabilities for RD0500, and we can project them through the alignment to where we think there will be base pairs in RD0260. So in the upper left-hand corner, I made a thermodynamic prediction. In the lower right-hand corner, we've transferred that to another tRNA. So we call that extrinsic information. And we, in Turbofold, incorporate this into an estimation of pair probabilities. And in this way, we can refine the pair probabilities where one sequence can inform another sequence. And so in Turbofold, the input is a set of multiple homologous sequences. In the first steps of Turbofold, we do pairwise probabilistic alignment of all pairs of sequences. And we estimate the base pair probabilities for each sequence using just thermodynamics. In the next step, we then take our pairwise probabilistic alignments and our base pair probabilities, and we calculate the extrinsic information for each sequence. We then feed that back into a new estimation of base pair probabilities for each sequence, and this helps us arrive at a consensus structure across the sequences. After we've done that, we can then take the base pair probabilities and calculate what's called a match score and incorporate that into the pairwise probabilistic alignment. So in that way, we can incorporate structure to refine the alignment. And so we then move back and forth, refining the base pair probabilities 3 times and the alignment probabilities 3 times. And then in the last steps, we can take the pairwise probabilistic alignments and generate a multiple sequence alignment. And we can also generate structures for each sequence. And so here's an example where this really pays off. So this was a benchmark done for RNase pRNAs. And so this uses multiple sets of 20 sequences where we selected them at random. And so to characterize the accuracy, we look at 2 things. We look at the sensitivity, which is the fraction of known base pairs correctly predicted. And we look at the positive predictive value, or PPV, which is the fraction of predictions that are correct. So if we're missing pairs in our models, that lowers our sensitivity. If we start predicting wrong pairs, that lowers our positive predictive value. And so here I've benchmarked Turbofold against Locarno, which is a method that simultaneously folds and aligns multiple sequences. RNA olifold, which is a method that takes a fixed input alignment and then infers the conserved structure given that fixed input alignment. And then max expect, which is a single sequence method from our software. So this is a negative control. And what we can see is that the stars indicate that Turbofold has significantly better performance and sensitivity compared to these other methods. And the stars here indicate that Turbofold also has higher positive predictive value with significance. And so the reason that Locarno doesn't do as well as Turbofold is Locarno has a very rigid model for what it considers a conserved secondary structure. So it's not as easily able to consider inserts of new base pairs as Turbofold is. RNA olifold doesn't do as well as Turbofold because RNase pRNAs can evolve quite a bit at the sequence level. So making a fixed input alignment doesn't correctly reflect the structured alignment. And then max expect is a single sequence method. So we hypothesized we would do better than a single sequence method. So we've also worked on some important extensions of Turbofold. So we can identify conserved non-canonical pairs. And we published that in PLOS Computational Biology. And in this work, we use Marc Parisian's and Francois Major's energies for quantifying the stability of non-canonical base pairs. We also demonstrated that chemical mapping by shape is synergistic with looking for conserved structures. And we published that in the Biophysical Journal. And so Turbofold is a part of our RNA structure software package, which is an open source package for RNA secondary structure prediction and analysis. It's available for download from our lab website. It uses a graphical interface in Java, which works on Windows, Mac, or Linux. We also have web servers, command line interfaces, and we also have C++ classes for programmers. These also have Python bindings. So lately we've worked on making our algorithms faster. And this has been a nice collaboration with Jiayan Wang from Oregon State University in computer science. So this is Liang, and this is his team, Cezanne Li, He Zhang, and Liang Zhang. And so Liang has a background in natural language processing. And so he's been able to take algorithms he developed for natural language processing and bring them to RNA folding. And so we worked together to linearize our RNA folding algorithms. And so we say that our traditional RNA folding algorithms are order n cubed. And what that means is if you double the length of the sequence you want to consider in a calculation, it takes 2 to the 3rd power or 8 times as much computer power or computer time. In a linearized algorithm, doubling the length of the sequence only takes twice as much computer time. And so the way this is done is also with dynamic programming, but what's used is what's called beam pruning to reduce the search space. So when the search space is reduced, that makes the search approximate. So we're no longer guaranteed to get the optimal solution, but it turns out we found that the prediction accuracy is actually slightly better. And what that tells us is that there are features of RNA folding we just don't understand yet. And so here's the payoff demonstrated in this plot. So this is the log of the computer time as a function of log of the sequence length. And so Vienna RNA fold is a method that can predict secondary structures and is quite fast, but it scales with the traditional n cube. And so that makes this curve quite steep. But linear fold, which is our linear folding software, scales linearly, and out to 10,000 nucleotides takes less than 2 minutes to predict a secondary structure. And so, so then we turned our attention to linearizing Turbofold. So Liang's team was able to linearize Turbofold, and this is the payoff for a 5-sequence calculation in Turbofold. So this is a plot of runtime in minutes as a function of log of the average sequence length. And so what you can see is that our traditional Turbofold with the cubic scaling really explodes in the amount of time needed as the sequences get longer. So we're able to look at conserved structures for ribosomal RNAs quite easily. But if we wanted to look at 10,000 or 30,000 nucleotide sequences, the time really explodes. But linear Turbofold is able to run out to 30,000 nucleotides in less than an hour. And so that enables us to look at whole viral genomes like SARS-CoV-2. So this is a prediction using linear turbofold of the conserved secondary structure of SARS-CoV-2. And in this calculation, we use 25 genomes. So 16 SARS-CoV-2 genomes and 9 SARS-related genomes. And so one important aspect of this is that our predictions recapitulate what was known by experimental mapping work and also manual comparative analysis of conserved stems in the 5' end and the 3' end of SARS-CoV-2. What's really nice, though, is that we actually predict an interaction between the 5' end and the 3' end of the virus. And that's novel for computational work, but it turns out Omar Ziv and Eric Miska published a bioRxiv paper demonstrating by cross-linking this interaction. So we're able to recapitulate this experiment. In red, we indicate positions of compensating base pair changes. And so I'm only showing the very 5' end and 3' end of the virus, but there's another 29,000 nucleotides of secondary structure we're able to make predictions for. And so using this predictions, we're able to identify unpaired and conserved regions for the design of RT-PCR primers or siRNAs or antisense DNAs to target SARS-CoV-2. We can also identify conserved loops for design of small molecule drugs. And this work is also then independently supported by compensating base pair changes. So in Turbofold, we don't use any term to encourage compensating base pair changes, but when we observe them in our modeled structures, they support our structures. And so we've posted a preprint of this work on bioRxiv. So in summary, Turbofold iteratively solves the alignment and folding problem to automate comparative sequence analysis. It is more accurate than other tools because it does not rely on a fixed alignment and because it can allow conformational flexibility. It's a part of our RNA structure package. And Jiayan Wang and his group have linearized Turbofold, making it possible to run on whole viral genomes. So I want to end by thanking Liz Leverens, who's our administrator in the RNA Center, for making this seminar possible. I want to thank my group members for their work, especially those who worked on Turbofold. So Yinghan Fu, Arif Harmansi, and Zhen Tan were former members of the group who worked on Turbofold. I want to thank Gaurav Sharma, who's been my close collaborator for over a decade now. He's in electrical and computer engineering. And I want to thank Jiayan Wang, who's now a close collaborator, who's in computer science at Oregon State University, and his team, Suzanne Li, He Zhang, and Liang Zhang for their work on linearizing the algorithms. And I want to thank my funding from NIH. And thank you for your attention. So Lynne, you're still muted.
Okay, thank you. And Dave will take questions now.
So I see in the question and answer, Todd Lowe asked, are there any other new folding insights in the rest of the viral genome? And so, so the answer is complicated in the sense that, yes, there's a lot to mine in the rest of the viral genome. In particular, we find conserved structures that could be good drug targets or could be good targets for siRNAs or antisense DNAs. And Austin Herbert asked, can you set a reference sequence with Turbofold? And if not, how do you recommend parsing the Turbofold output of thousands of sequences for the greatest change in structure from the consensus? That is an excellent question. So in the sense, you can have a reference sequence where if you know the structure or you know some aspects of the structure by experimental mapping, for example, you can require that structure to be in that reference sequence. And then the question is, how do you then parse the Turbofold output? I can imagine two important ways of doing that. So one might be just counting the number of base pairs different compared to your reference structure. The other thing you could do is calculate the root mean squared deviation of the base pair probabilities compared to the reference structure, which might be a little bit more sensitive. So Li Ji asked, how many conserved sequences are required for prediction? And that is an excellent question. So if you choose the right sequences, you do not need very many. And what I mean by that is if the sequences are in kind of a sweet spot where there's enough sequence identity to get reasonable sequence alignment, but enough difference to require or to make it hard to predict too many spurious base pairs, then like a handful of sequences, like 5, would already be a dramatic improvement over one sequence. For SARS, where there's much less sequence variation, it was much more beneficial to go out to like 20 sequences, and there may be more to gain by going even farther out. So Ashwin Kumar asks, have you looked at the kinetics of folding and stochastic nature of non-equilibrium thermodynamics for RNA structure prediction? So that's an excellent question. So for the most part, we've focused our predictions on equilibrium predictions. So we do know for some specific RNAs that there can be kinetic effects. So in other words, an RNA could get stuck in a local minimum and not be able to find the global minimum. And in those cases, Turbofold can overcome the limitations of just focusing on thermodynamics, because by comparing multiple sequences, we know we should get the biologically relevant structure. So Niels Walter asks, what about including AI? And so he points out Riju Das's recent science paper comes to mind as an example applied to 3D structure. So we've done a little bit of work looking at AI. And of course, I look at all the papers looking at AI. And maybe the short answer is that artificial intelligence or neural networks have a lot of promise. In secondary structure, I would say generally we see that the current methods are not very transferable. And so, so a number of papers have appeared that tend to train and benchmark on closely related sequences. And so not enough work has been done looking at sequences that are more distantly related. Nato Hori asks, does the slightly better accuracy when some approximation used indicate that Turner's rule is not working perfectly in some cases? And I would say the short answer is yes. So we know that the Turner rules, or our nearest neighbor models, don't even reflect everything that's known experimentally today about RNA folding stability. And so we're collaborating with Doug Turner and Susan Schroeder to improve the nearest neighbor rules. And so Turbofold is one way that we can improve accuracy by trying to overcome some of these limitations. And so Todd Lowe asks, how does Turbofold do with ribo switches? Is it confused by 2 competing conserved folds? So that's also a great question. And so one of the strengths of Turbofold is that it works probabilistically. So the hidden Markov model alignments are probabilistic, and the base pair probabilities are probabilistic. And so, so although we've not looked carefully at ribo switches in detail, Turbofold should be able to do well, because if you had competing structures, the pair probabilities should reflect those 2 structures, and we should be able to observe both in the predictions. And then Syed Tarabi asks, what do you recommend as the best primary sequence aligner? Yeah, that's a good question. I'm probably not the best person to answer that. There are probably other people with more expertise, but there have been some nice benchmarks that have been done benchmarking different alignment algorithms for RNA sequences.
Dave, have you ever examined the ability of Turbofold to address how an RNA binding protein might influence structure?
Yeah, that's a great question. So we have not done that with Turbofold. So we have, yeah, we just haven't done that yet. So, I mean, there are ways we could incorporate that information if we knew that like a protein could fix a secondary structure, we could fix that in the calculation. Or if a region had to be unpaired, we could account for that. But we've not done a lot of calculations with Turbofold and proteins.
Well, we can talk more about that because we have some examples for you. So remember, you can go to Discord and ask Dave and Jenna any further questions that you might have. And I want to give a shout out to the NIH NCI initiative talks. That will be September 22nd. They are the next series of talks, and they will be given by Astrid Haas, I hope I'm saying that right, and Jay Schneekloth. So thanks, everybody.